Cells communicate through direct contact, short distances, and over long distances. So, a lot of this is going to be illustrative examples of how cell communication happens. Now, cells can communicate cell to cell through direct contact. And in fact, sometimes it's this direct contact that prevents cell division because there's no space for more cells. So, not all of your cells in your body are touching, but some places in your body are. At these direct contact locations, there can become gap junctions where membrane channels are shared between nearby cells, which signaling molecules can transport through those. Now, plants have a very similar situation with those plasmodesmata. So these breaks in the cell wall where cells are touching each other, and again, signaling molecules can go through there, and including things like plant defense communication. So, talking about defense, um, when we look at the human immune system and inflammatory response, we can see communication happening through direct contact and through over short distances. So if you have an injured area in the body, mast cells will release histamine, which will cause vasodilation. We'll also have these macrophages that will be eating pathogen and releasing cytokines. So these cytokines are signaling molecules just like the histamine, and these cytokines are going to call in other help to the area, including bacteria slayers like neutrophils. So another way of looking at this is through this demonstration. Here we have mast cells, here we have macrophages, we have things traveling through the blood like neutrophils and eosinophils. All of these B cells, helper T cells, killer T cells, these are all white blood cells or leukocytes. So as we progress here, we have cellular damage, and the mast cell is going to release histamine, which is going to cause vasodilation. This leakage of white blood cells into the area can cause some consumption of, and eating of the pathogen. The macrophage will also do a little bit of eating, and they will present antigens on their surface where direct helper T cells can come in direct contact and send out interleukins or cell signaling molecules, cytokines, to call in extra help. The cytotoxic T cells, the killer T cells, will monitor our own cells for infection, and if they notice abnormal antigens, they can kill and destroy our own cells before they can become factories for producing more viruses or bacteria or more pathogen. So again, how that works, we have this direct contact with our cytotoxic or killer T cell and our own cell. So this could be cancer, but it could be a virus-infected cell. So as it comes in with this antigen fragment that it interprets as abnormal, at this direct contact, the cytotoxic T cell will cause this vesicle of perforin to create a hole in the target cell. And these granzymes will come in and cause the cells to go through apoptosis or programmed cell death. And therefore, the virus can't use the target cell as a factory to make more copies of itself. So again, we have this place where the helper T is calling in the B cell, and the B cell is going to make contact with the pathogen and again present a little antigen. That helper T cell is going to come in contact once it's been activated and the B cell has become activated to send messages to this B cell to become plasma cells. And these plasma cells shoot out antibodies in great amounts, which then can neutralize, clump, or agglutinate the pathogen so that other small macrophages like eosinophils can come and consume the pathogen. And again, through all that, we get memory cells and immunity. But again, that immune system will communicate through short distances and through direct contact between cells. Another version where we don't quite touch, but we have short distances, is the idea of neurotransmitters. So this at the synapse, we have a cleft, a little gap here. So as the action potential is heading towards the axon terminal, eventually calcium ions will go in through voltage gated channels, which will cause the release of these vesicles of neurotransmitters. These ligands will cross the cleft to the next cell. And it's at this next cell where they land on the ligand gated channel, and that ligand gated channel causes it to open at this receptor here, and the sodium ions can rush in, which then creates the next action potential and sends the communication further on. But again, this isn't quite direct contact, it's over short distances. Another short distance example would be with bacteria and quorum sensing. So bacteria are naturally going to produce these autoinducers. And there are certain things that bacteria do that they only do once they get to a great enough density. So this idea of quorum sensing, the more bacteria that exist, the higher the concentration of these autoinducers. When these autoinducers get to a high enough concentration, they communicate back to all of these bacteria to do some unique things. Some of it could be bioluminescence, but a lot of times we have this biofilm that they start creating, but they only start creating them once they have enough bacteria density. So those biofilms, it can be like this extracellular matrix that goes around the outside, which is going to prevent antibiotics from being able to actually at least slow them down and prevent them from reaching the bacteria. There's also things where they don't start triggering the creation of toxins or dangerous enzymes to their host until they get enough of them together, density, to actually create a problem. So a single bacteria might not start releasing toxins, but once you get to this quorum sensing level and you have this short distance cellular signaling, that they'll actually start making the toxins that could kill a host. So as we start going from short to longer distance, more examples here. If a leaf of a plant is starting to get eaten by an insect, the leaf itself will notice that damage and will release this cysteamine. And it's going to communicate to the leaves nearby, but it'll also communicate to the leaves throughout the plant. And this cysteamine is going to land on a receptor, which is going to trigger these different chemicals, these defense chemicals, which we're going to go through signal pathways, which we'll get to, and that's going to activate certain genes within the nucleus. And it's these proteinase inhibitors that end up causing the plants to be like poisonous or really foul tasting. And those genes that make those inhibitors aren't created constantly by the plant. A lot of times they're released in response to being damaged by insects. And even some of this can evaporate off the surface and trigger other plants nearby to go through a similar process. So the classic human example of communication over long distances is the endocrine system, where we have steroid and non-steroid hormones. So again, we have a lot of stimulating or tropic hormones that are released from our brain to other glands in our body. Some glands sense chemical levels on their own and monitor the blood and can release hormones through the blood to target cells throughout the body. And there's lots of examples. The pineal gland has melatonin. A lot of these you've heard of, like, and maybe antidiuretic hormone. That example is going to come up later. Insulin, glucagon, testosterone, and we're going to get to estrogen and testosterone and some of our FSH and LH in a second. But we also have adrenaline, epinephrine, norepinephrine, thyroxine from your thyroid glands. Calcitonin and parathyroid hormones are going to control your calcium levels. But again, this stuff gets released through the bloodstream and can affect things in faraway places. So we have, our pituitary gland, which has an anterior and posterior pituitary that's located up in our brain, but it can have target cells far away from the body. for example, in the kidney tubules, in the uterus, and in the mammary glands. When we start looking at again, at the other side of the pituitary gland, we have stimulating hormones that are going to affect our thyroid gland, our adrenal gland, again, mammary glands, but growth hormone is going to affect bones and tissues. And again, FSH and LH are going to affect the ovaries and the testes. So as we look at this, again, the anterior pituitary is going to release LH and FSH that are going to go to the testicles, which is going to ultimately produce testosterone. So as we look at this, we start with hormones. So at puberty, FSH, follicle stimulating hormone, and LH, luteinizing hormone, is released. And they go to they have different target cells. So the FSH, again, travels through the blood down to the testicles to the seminiferous tubules. And we have these spermatogonia cells that are receiving the FSH. The LH is going to these cells in between the tissues, these interstitial cells, which then triggers the release of testosterone. So again, this FSH triggers some mitosis, and then they go through meiosis. And ultimately, it's this testosterone that's going to help trigger the maturation of a tail and loss of excess cytoplasm so that sperm can drop into the lumen of the tubule and ultimately be able to help a male produce offspring. So on the women's side of things, we again still have FSH and LH, but now we're focusing also on estrogen and progesterone. Instead of the testicles, we are looking at the ovaries. So again, before birth, we have these primary follicles from these oocytes, which are going into the ovaries, but it's the FSH that causes this follicle stimulating hormone that causes this maturation of and developing of this follicle. So when LH hits the first time, luteinizing hormone, this developing follicle is going to secrete estrogen, and that estrogen is going to prep the endometrium or the lining of the uterus to become filled with blood vessels. Again, this second surge of LH is causing the actual ovulation, where that secondary oocyte is going to go into the fallopian tube for possible fertilization. So after you have this ruptured follicle where the secondary oocyte goes into the fallopian tube, this ruptured follicle becomes the corpus luteum. And again, the estrogen is prepping this lining, and the progesterone that's released from the corpus luteum is also triggering this to secrete nutrients to sustain a fertilized embryo. And again, this corpus luteum doesn't last forever. So if we do have implantation, ultimately the placenta takes over with continuing to sustain the pregnancy by secreting progesterone, which prevents contractions and actually kind of dulls the woman's immune system to not reject the developing embryo. So again, all of these are lots of examples to understand that cells communicate through direct contact over short distances and over long distances.